Hi, I'm Minyong and Jim, a research scientist at NEB.
I'm Jim Samuelson, a senior scientist at NEB. We're going to talk about our recent work published by Nature Communications. The title of the paper is An Engineered High-Affinity Fbs1 Carbohydrate Binding Protein for the Selective Capture of N-glycans and N-glycopeptides.
The goal of the study was to develop a specific, efficient, and unbiased method to enrich intact N-glycopeptides to facilitate large-scale N-glycoproteomic studies. Protein glycosylation is one of the most common post-translational modifications. It plays important roles in protein folding, trafficking, cell adhesion, and signaling. It is common for specific glycoside to have many different glycan modifications. This heterogeneity of glycosylations may be affected by cell physiology and disease states. Therefore, it is a potential biomarker.
There are primarily two types of glycosylation: O-glycosylation, in which the glycan is attached to either a serine or a threonine residue, and N-glycosylation, where the glycan is attached to an asparagine residue. We focus only on N-glycosylation in this paper. To determine N-glycosylation heterogeneity, a glycoprotein sample is typically digested with a protease such as trypsin. The resulting peptide mixture contains a very small fraction of intact N-glycopeptide and a very large fraction of standard peptides. Compared to standard peptides, intact N-glycopeptides are difficult to detect by mass spec due to the hydrophilic nature of the glycan, which causes low ionization efficiency. Therefore, a specific unbiased enrichment method is required for intact glycopeptide analysis.
To achieve this goal, we searched for a protein that can bind to the core motif of N-glycans rather than the variable regions. We identify a protein named Fbs1 which can recognize and bind to the core motif, compared to lactin enrichment, in which lactins bind to the variable region of glycans. Fbs1 offers more specific binding to N-glycans. We took advantage of this inherent property of Fbs1 to establish an N-glycopeptide enrichment method. In this figure, wild-type Fbs1 and Fbs1-GYR bind to a series of N-glycopeptides appended with different N-glycans. Wild-type Fbs1 shows significantly biased binding, while Fbs1-GYR binds with similar affinity to the 4 glycopeptides. Next, we applied the Fbs1-GYR mutant to enrich and identify intact N-glycopeptides from human serum using LC-MS. Furthermore, we compared our method to the standard lactin enrichment method. With the same input of tryptic peptides, 2.2-fold more N-glycopeptide spectrum were identified using Fbs1-GYR relative to lactin enrichment. And 7-fold more spectra were identified when compared to pre-enrichment samples. Following Fbs1-GYR enrichment, 66% of total spectrum were assigned to N-glycopeptide. Our data demonstrate that Fbs1-GYR enrichment greatly improves the identification of N-glycosylation heterogeneity. To be more specific, Fbs1-GYR enrichment results in an average of 5.4 glycan types per glycoside and 13.4 spectrum counts per glycoside, a 3.3-fold improvement over the values without enrichment. The complete dataset from the heterogeneity analysis is presented in the supplementary data of the paper. Here shows an example of heterogeneity analysis for a single potential disease biomarker, human complement C3. Fbs1-GYR enrichment clearly enabled identification of more glycan types and spectrum in the 3 glycosides, relative to the pre-enrichment and lactin enrichment samples.
In summary, when applied to biological samples, Fbs1-GYR enrichment results in deeper coverage and a clear snapshot of the heterogeneity present at each glycosite. Therefore, we expect that large-scale N-glycoproteomic studies will be positively benefited by our work. For more information, please visit the Nature Communications website to find our open access publication.